this is Rebecca Seal, professor and pain researcher at the University of Pittsburgh. Welcome to a new feature of the Pain Research Forum called Pain Spotlight, Voices Shaping Pain Science. Developed and hosted by Drs. Gomes-Aragão and Beatriz Lima at Adjafre. Today's Spotlight episode is part 1 of sodium channels and the making of a field, featuring Dr. Michael Gold. Michael reflects on the discoveries that shaped modern pain neuroscience, from the identification of sodium channels as drivers of nociceptor excitability to the emergence of NaVs as therapeutic targets. This conversation bridges the history of pain research with the future of mechanism-based analgesia. Thank you for joining us, and we hope you enjoy.
Hello guys, my name is Isaac.
And I am Beatriz.
We are postdoctoral researchers at the University of São Paulo in Brazil. Also, we are early career contributors to the Pain Research Forum of the ISP, and today we have the pleasure of introducing Professor Michael Gold. Michael Gold is a professor of neurobiology at the University of Pittsburgh. Dr. Gold received his BA from UC Berkeley, his PhD from UCLA, and was a postdoctoral fellow with John Levine at UCSF. The focus of Dr. Gold's research is the neurobiology of pain. He has made important contributions to injury-induced plasticity nociceptive afferents and their contribution to the manifestation of persistent pain. Toward this end, he has employed an array of approaches, ranging from the study of isolated cells to the development of behavioral assays with which to assess the presence of persistent hypersensitivity, and more recently, the study of clinical populations suffering from persistent pain. Dr. Gold is actively involved in the greater pain community. Michael, thank you so much for being with us today. Could you briefly share your journey and how sodium channels became a central focus in your research?
Well, thank you for inviting me to participate in this. I'm excited about the reboot of PRF and the growth of PRF. I guess I came to sodium channels via the question that I was asking at the time, which was, what is controlling the excitability of nociceptors, particularly the relatively rapid response to inflammatory mediators. Both human psychophysics data and preclinical data suggested that inflammatory mediators like prostaglandin E2 could produce an increase in the excitability, both the presence at a behavioral level, hypersensitivity, but at a neuronal level, an increase in the excitability of nociceptive afferents in the time course of tens of seconds. And so that suggested protein-protein interactions and suggested that the channels underlying that change in excitability should be dynamically modulated. And so I was, at the time, had been focused on potassium channels, in fact, because it's a much larger family of channels that has been shown in a variety of systems with a variety of different potassium channels to contribute to the dynamic regulation of excitability. I spent a lot of time first trying to isolate the different potassium channels in sensory neurons and then look for their dynamic regulation, predicting that I would see a decrease or suppression of a potassium channel. And after what was literally years of work, isolating these channels and looking for responses and not seeing anything, I sort of turned to what was another dominant channel in nociceptive afferents, which was this TTX-resistant sodium current. When I started studying it, it hadn't been cloned yet. It was cloned in 1996 by almost two groups simultaneously, Lakshmi Samswaran and Roche, and Arman Akopian, when he was still with John Wood, calling it SNS or PN3 independently. But it was really one of those aha moments of science where I was recording the currents, they're very stable, put on PG2, and within seconds you start to see an increase in the current. And it's still one of the most exciting days of my scientific career because, I knew immediately that this was it. This was the channel mediating this sort of rapid increase in excitability that inflammatory mediators are able to do or drive. and therefore contributed to this sort of inflammatory hypersensitivity that's seen immediately after injury. And that's sort of what set the stage for looking at those channels and at NaV 1.8 in particular.
Oh, yeah, that's pretty good way to start finding like something that's very most science starts, right? We find something, we believe in something that works, then that thing doesn't work, and we find other things that truly, really work. So it's really interesting, the start of your story. And Professor Gold, your studies on NaV 1.8 and other channels have shed light on how injury and inflammation dynamically modulate nociceptor excitability. So could you describe how the NaV 1.8 channel contributes to nociceptor sensitization after injury or inflammation, and how the regulation of this channel differs from NaV 1.7, for example?
Sure. So the 1.8, John Wood's group's shown it has phosphorylation sites, and those phosphorylation sites are able to change basically the gating of the channel. Sensory neurons are some of the biggest neurons in the body. The cell bodies are in the ganglia that run along the spinal cord, in the dorsal root, or above the neck in the trigeminal ganglia. They can be at least the low threshold afferents can be up to 1 meter long. And interestingly about sensory neurons is that they distribute different channels in different places. Different potassium channels are differentially distributed along the axons. Different sodium channels are distributed along the axons. NaV 1.8 happens to be one of these channels that's distributed. It's expressed in the cell body, so you can record even in vivo the currents in the cell bodies, but it also appears to be preferentially distributed under normal conditions to the terminals, both the peripheral terminals and the central terminal. So it's in a position to be able to contribute to that initial spike initiation. And so if you imagine what is referred to as a generator potential, this might be a piezo2 channel that's activated with mechanical pressure, or a TRPV1 that might be activated with an increase in temperature. Those channels will open. They'll enable a membrane depolarization. That membrane depolarization ultimately has to drive the emergence of an action potential. And that emergence of an action potential requires voltage-gated sodium channels. And so the biophysics then, how that voltage-gated sodium channel opens and closes, and the voltage at which that happens will influence the excitability of a nociceptive neuron. If that threshold goes down, the neuron becomes more excitable. If that threshold goes up, similarly, if there are more channels that are available to contribute, it's easier to drive that neuron to a threshold, or to the action potential threshold, and for an action potential generation. And so when you've increased the excitability of the neuron by dynamically modulating the channels so that those channels open more quickly or more easily or at lower thresholds, you've basically dropped the threshold, and that accounts for the drop in mechanical threshold or thermal threshold that we observe in the presence of inflammation. And because they're in the terminals, they're in that position to be able to respond to mediators release, whether it's from immune cells or other damaged tissue, to contribute to that initial increase in excitability. How they contribute at later time points can change. Many types of pain processes involve the ongoing release of inflammatory mediators like PGE2. So there's this sort of ongoing drive for the sensitization of these channels that might contribute to why a non-steroidal like ibuprofen works for a knee, an inflamed knee, over time. But depending on the type of injury, the channels will also, you can start to see a change in expression. So you get an increase in channel current density, or the number of channels present, and that can contribute to an excitability. And you can get a change in the distribution, which seems to differentiate the response to nerve injury versus the response to inflammatory stimuli. NaV 1.7, the dynamic regulation of 1.7 hasn't been nearly as well studied compared to NaV 1.8, just in terms of the changes in the biophysical properties of the channel, if you will. But Steve Waxman's group and Suleiman Dibhaj have shown very nicely that there's modulation of NaV 1.7 largely in terms of trafficking. And so how the channels are trafficked along the axon seems to change, and changes in a temperature-dependent way, which in their hands, they argue, contributes to, say, some of the temperature sensitivity that is observed in some of the channelopathies, like those associated with the gain-of-function mutations that drive erythromelalgia, or primary inherited erythromelalgia.
How do you see inherited channelopathies and injury-induced plasticity interacting? Do you think these mechanisms are convergent in the pathways that lead to chronic pain, or do they operate via different processes?
Yeah, so that's a great question. The channelopathies, by definition, so these are basically mutations, if you will, for lack of a better word, and no pejorative term intended, are alterations in the channel sequence because of mutations in the gene. And those are generally referred to as gain-of-function mutations or loss-of-function mutations, based on what they do to the properties of the protein. In the case of sodium channels, the gain-of-function increases the functioning of the sodium channel. Many of the gain-of-function mutations associated with primary inherited erythromelalgia, that is in NaV 1.7, primary inherited erythromelalgia, paroxysmal extreme pain disorder, are associated with increases in current density, shifts in the voltage dependence of inactivation and activation that result in an increase in the ability of that sodium channel to contribute to action potential generation. And that's what is thought to, at least in part, contribute to the phenotype. My view, however, of the channelopathies tends to be a little contrary to the I don't know, the weight of at least how other people view it, and that is that, at least in my mind, what the channelopathies are doing, particularly for channels like NaV 1.7 that are expressed relatively early in development, is that they're not only influencing what the channel does sort of in its adult state, how it's functioning in a mature nervous system, but they influence the development of the nervous system. And consequently, there can be compensatory changes that occur as the nervous system develops that can be as important, if not more important, to the phenotype in the adult as the sort of properties of the mutated channel. And I think the gain-of-function mutations with NaV 1.7 are a great example of that, where this is a channel that's distributed throughout the body. But with primary inherited erythromelalgia, the pain that the patients experience is primarily restricted to their hands and feet and is primarily associated with changes in temperature. In contrast, paroxysmal extreme pain disorder is localized very specifically to a particular part of the body, the anal sphincter, during development. And if you live long enough with paroxysmal extreme pain disorder, it'll migrate to pain in the jaw and then pain in the eye. And that distribution, in my mind, cannot be simply the reflection of a channel that is increasing the excitability of the nervous system, but has to be reflecting the interactions of that channel with the other channels that are present in the neuron, how the system is wired up, and how the different neurons are connected. And interestingly, in that context, particularly with paroxysmal extreme pain disorder, that you don't have pain elsewhere in your body really suggests that even though these channels are hyperexcitable, the nervous system is adapted to the point where they don't drive nociceptors being more excitable, so that you have pain distributed throughout your body. It really is very specific to this sort of combination effect that I would argue is largely because of the interaction between the channel and the development. So interpreting the channelopathy data really has to be done, in my mind, through the lens of what is the changes in the protein structure that influence its just biophysical properties, potentially distribution, et cetera, but also how it's influencing the development of the nervous system. And that's in contrast to the dynamic modulation of a channel in the adult nervous system that is probably involved in just sort of the dynamic changes in excitability that we might have experienced during a stressful event where you've got, the release of systemic levels of catecholamines and other sorts of things. Those mediators are able to act on receptors that are going to dynamically regulate all the channels in the neuron throughout the nervous system, and therefore influencing sort of the level of excitability, if you will. So you're hypersensitive in some conditions and you're less sensitive in other conditions, simply because of that sort of dynamic regulation that you see. I think those are the two biggest differences in my mind. One is how the system is quote unquote normally wired to respond to sort of fluctuations in mediators versus how the system is potentially aberrantly wired, folded on top of how these channels are not functioning normally as well. So you sort of have that combinatorial effect in the channelopathy.
Great. Yeah. For final question of this section about sodium channels and pain, I want to ask you that given the heterogeneity of nociceptors and their ion channel profiles, which are multiple, as you have described prior, how does this complexity influence our ability to develop targeted therapies? How do you see targeted therapies for this multitude of channels?
Right. Yeah. So that's a great question. And it sort of speaks to, I think, the difficulty in terms of developing therapeutics in general. And so, why has it taken us so long to develop novel therapeutics? Prior to the approval, at least in the US, of suzetrigine, this Vertex compound, the 1.8 blocker, that was really the first new treatment for pain outside of the space of migraine in many, many years. And that's despite, this very concerted effort to understand that, what controls the excitability, the tremendous work that Artem Pataputian and Dave Julius had done, their Nobel Prize winning work in terms of identifying these transducers, that fundamentally that increased understanding has failed to develop, result in more therapeutics because of how complex the system is. It's one of those sort of classic, I enjoy science because the more you learn, the more complex it seems. It's frustrating for people who want therapeutics. If the study that you've just completed doesn't raise a whole host of new questions, then, there's something wrong with the study. And that's great for science. It's not so great for therapeutics. And so the system is complex. And you alluded to that with the question of that there's these many nociceptors that are doing different things. I think in terms of therapeutic development, identifying sort of, pinch points or points of convergence, if you will, is a way forward. And that's why I still have hope for NaV1.8 as a therapeutic target, because there's a whole host of different inflammatory mediators, PG2, IL-1 beta, IL-6, that have been shown to converge on NaV1.8 as a target that is then contributing to the change in excitability. So rather than having to use a COX inhibitor and antibodies for each of those different mediators, blocking NaV1.8 enables you to sort of short circuit, if you will, that myriad of inflammatory mediators that are released in the context of inflammation. So those pinch points are good, but as you also alluded to, the relative contribution of those channels varies among subpopulations. And depending on the site of injury, the nature of the injury, the timing after injury, all the available data that we've collected would argue that the specific channels, the specific neurons, the specific combination of mediators is going to be context dependent. From a therapeutic perspective, and not only development, but the prescribing of therapeutic perspective, is that we have a hard time yet identifying where the nervous system is in the sort of the trajectory post-injury. Is it that sort of protein-protein interaction component? Is it now the protein expression? Is it that we've gone from a contribution of, C polymodal nociceptors to a different population that's contributing? That sort of dynamic contribution, I think, was beautifully illustrated by some of the early work out of the Hopkins group, recording from non-human primate nociceptors, where if you just look at the time course of the response to heat, Psychophysically, you apply the heat to the forearm of a human, and they report that it hurts at a 7 out of 10. And that report will be relatively constant across the 30 seconds of a stimulus. But if you're recording from the neurons that are innervating that skin, the A deltas will turn on first, and then they turn off, and then the Cs will kick in a little bit later. And so you have this relative contribution of fibers that is changing over just that 30-second time frame. If you expand that out to tens of minutes, to hours, to days, the available evidence that we have says that can also change dynamically. That's certainly true in the context of nerve injury. Inflammation that may not be true, but the neurons themselves are also changing, right? There's all kinds of plasticity that has been very well documented in these nervous systems. So the trick is really to identify the population that is contributing at any given point in time. And so we sort of have a combinatorial problem of not only developing these novel therapeutics, but developing the ability to identify where any individual is in their trajectory. And I think we're getting better at that. A lot of the work that, say, Ted Price and Rob Giroux are doing in terms of the mRNA expressed in sensory neurons and that others have done in the context of injury and how that changes, I think are going to be important for at least pointing us in the direction of identifying those changes. But we and others, Ted as well, Ted Price, have been looking at local translation. And you're going to miss that by looking at the cell body or the nucleus. And if the proteins that are relevant for the excitability aren't, the expression or the translation isn't happening at the cell body, but it's actually happening out in the axon. I think the tools are getting better, right? We are developing ways of assessing less invasively what's going on in a human over time, but we still have a long way to go in that context, and being able to sort of match that timing is going to be key. The complexity for me is fascinating, but I tend to be a splitter and not a lumper. And I really look at, a lot of our work has been comparing dural afferents to muscle afferents, or muscle afferents to visceral afferents, or skin to this or to that. And all that data suggests that they really are different. What controls their excitability and how they respond to stimuli is different simply by that target of innervation.
Despite decades of research, sodium channel blockers for pain have struggled to deliver on their promise. And recently, we have seen that the case of suzetrigine have emerged as a hopeful candidate. So I just want to ask you a few questions in regards to this matter of developing therapeutics. Your work highlights how factors such as sex, immune environment, and injury type modulate pain pathways. Do you think highly selective NaV 1.8 blockers can effectively address such complexity in pain states?
The short answer is yes, but conditionally, right? Yes, but in the specific context in which that channel is playing a dominant role. And so, at least from my perspective, part of the frustration with how Vertex went about pursuing the clinical efficacy of suzetrigine is they targeted postoperative pain as their primary indication. So the abdominoplasty and the bunionectomy were the two indications, or the two sort of surgical procedures that they focused on. The only preclinical data that was available in the context of the role of NaV 1.8 in either inflammatory or neuropathic pain or postoperative pain was negative for postoperative pain. It was Abbott, Joshi et al. that published that study. They used siRNA to knock down 1.8, and they had a nice attenuation of hypersensitivity associated with nerve injury. They had a nice attenuation of hypersensitivity associated with inflammatory stimuli, but they didn't really see an effect on the hypersensitivity associated with postoperative pain. I could speculate on why that might be the case. They still saw some effect, but it's relatively small. But I think there are other conditions in which 1.8 could and likely does play a much larger role, and therefore the effect size of the drug is likely to be bigger. Conversely, I think there are other pain syndromes for which the contribution may be significantly less, because there are other channels that are playing a much more dominant role. And so again, as we were just talking about, I think the key is going to be identifying those patient populations, where they are in their trajectory, that they're going to be most responsive to a 1.8 blocker. And I think there will be patients who are very responsive to suzetrigine, to NaV 1.8 blockers, but there are also going to be populations that are far less responsive.
I see. And do you foresee future like of a personalized pain medicine where genetic and electrophysiological and clinical profiles could guide NAV channel targeted therapies, and what challenges remain?
And I think personalized medicine is the extreme of that sort of splitting, right? That is that we all, each of us personally, require a therapeutic that is tailored to our specific needs, our history, our age, our sex, other factors, our specific type of it, et cetera, et cetera. And so I think those sorts of platforms are ultimately going to be how the future, how we address these complex problems in the future. The driving force behind at least the first generation of those platforms was really genetics and the assumption that all phenotype is a reflection of, your genetic background. In this lecture that I give on why we don't have therapeutics, I have a section called the hubris of the geneticist, which is that, this view that all we have to do is identify the gene and then we can solve the problem. And I think that the available evidence would just underscore again and again that is not simply the case, right? I mean, you need a lot more than the genetics. It isn't to say the genetics aren't going to help you. But, your history, your sex, these are all dynamic influences that interact with, not just what genes are being expressed, but how things are being post-translationally modified, et cetera, that's going to influence the phenotype. And so I think fundamentally what is needed is that personalized platform, but that personalized platform is going to involve far more than just genetics, right? It's going to be psychophysics. It's going to be, a solid assessment of where someone is in their sort of psychosocial framework. It's going to be a detailed history. The microbiome, diet's going to be important. And so the people who are collecting those kind of data in the U.S., there was a big study to study low back pain. We called it Backpack. And that was a sort of collect all kinds of data. It was a psychophysics, psychometrics, imaging data, the spine, some ecologically relevant variables following people over time. And that detailed assessment, microbiome, that detailed assessment on top of genetics and everything else enabled the subpopulation of multiple types of back pain. Are we at the point of personalized with that framework? Maybe not, but we're getting a lot closer. But in my mind, it speaks to sort of the volume of the data that is required. And this is where, I think AI, other sorts of things could be really useful of coming up with strategies to address these volumes of data to sort of point through, what might be the phenotypes that would be particularly responsive to X, Y, or Z.
So, how important is it to validate pharmacological targets in human tissue before advancing therapies? And in your case, as an American researcher, do you think it is hard to have access to human samples in your university or in your state for validating your findings?
Right. Well, so your first question is a softball one. So I've been working with human tissue for a while, and so I'm at least convinced myself that human tissue is an essential step. How channels function, how proteins function in their native environment is going to depend on the native environment. I think simply using heterologous expression systems, again, going back to this lecture that I give on the barriers to translation, there's very good data to suggest that even the cell type that you're heterologously expressing your protein in influences the properties of the protein. Volscicated sodium channels in particular with, tens, if not hundreds, of post-translational modification sites speak to a protein class that is going to be dependent. Its biophysical properties, pharmacological properties are going to be dependent on the native environment and how that protein is post-translationally modified. A classic example, again, going back to some of Abbott's earlier work, was their development of the first NaV1.8 selective blocker, A80. And that compound they showed had, 2+ orders of magnitude selectivity for 1.8 versus the other sodium channels. You look at those channels present in a native system, and you lose more than an order of magnitude of selectivity of that protein. Same target, but clearly the pharmacology changed in the native environment. So the short answer is, I think it's critical. And if you want to ultimately translate to humans, I think before you start a phase 1 clinical trial, which is what they call them in our country, of, this is first in human studies, just looking at toxicity. Those are very expensive studies to run, as you might imagine. You're looking at a drug that, hopefully is safe and hopefully isn't going to hurt your patients, but just testing those in people has a lot of appropriately regulatory burden associated with it. So it is potentially easier and cheaper to test human tissue if you can get it. So the if you can get it is the second part of your question. Here in the US, we have a very vibrant organ donation program. And the country is carved up by organ procurement organizations that essentially have a geographic area for which they are responsible. And across the organizations, they share the lists of who's on, who's waiting for an organ, the genotyping, and all sorts of other details about the organ types and who's going to be a potential donor. That process is very well established. In this country, if you are 16 years old, you can get a driver's license, and part of the driver's license process is, do you want to become an organ donor? And so, part of your sort of baseline ID is, if you happen to be in the wrong situation at the wrong time, you're willing to have your organs given up for donation. Organ donation is different, though, than using your tissue for research. And that requires, at least in this country, an additional level of approval. And the next of kin need to approve the use of tissue for research. And that's not an easy ask, as you might imagine. How we've treated particularly minority individuals, it's not a great history. The Tuskegee syphilis trial is a great example of just this went on for till 1974? Where they were basically doing this experiment on people who didn't give their consent, letting them be basically infected with syphilis. So not surprisingly, there's cohorts, particularly what are considered minority individuals in this country, who are skeptical about organ donation. And so, we've been studying tissue for almost 10 years now. And the proportion of minorities in our database is not at all reflective of the population. And again, I understand why, but we as scientists have a huge barrier to overcome in terms of restoring that confidence of everybody in research and convincing people that allowing scientists to use their tissue. The story of the HeLa cells is another great example. For lack of a better word, the abuse of, somebody's giving, in this case, it was tissue that was taken even without consent. I think human tissue is important. If you can get a collaboration going with the organ procurement organizations, you can get access to tissue from organ donors that, for all intents and purposes, is relatively healthy and provides a really powerful tool to look at tissues. It's going to depend on where you live in this country to how easy the procurement organization is to work with. But in general, many of them are very easy to work with. The other source of tissue is from the OR, and many different types of tissue that are removed as part of surgical procedures are discarded as medical waste. And so we've looked at tooth pulp, we've looked at knees. We've looked at a lot of tissue that is discarded as medical waste that can still be studied for the nerve content, for changes in normal physiology, changes in function. Dave Bulmer's group in the UK has really pioneered the study of human colon and looking at the innervation of the colon and those properties. So that's another source of tissue that I think if you're living in a country where organ donation isn't a key sort of ethos, isn't as well developed, that getting access to who's controlling those isn't as easy. If you're working in an institution that's associated with a hospital, that is generally an easier path to go by. And at least our institutional review boards, that is the boards that approve the use of human tissues, that's a relatively easy approval to get because you're not doing anything but just collecting tissue being removed as part of a procedure. And that's in part how Ted Price and his colleagues in Texas have been getting surgical specimens of DRG to study to complement the organ donation.
So now moving on a bit from sodium channels to look at the whole picture. Pain, we know, is a system level phenomenon with peripheral and central components which are deeply intertwined. And so we would like to ask you, Michael, your studies in migraine and visceral pain emphasize neuroimmune crosstalk and hormonal influences on that. And how do these systemic factors shape nociceptor function and sodium channel regulation?
Yeah, so that's another great question. There's a journal, sort of the U.S. Pain Society's Journal of Pain is the Journal of Pain. And before the Journal of Pain became the Journal of Pain, it was the Pain Forum, not the Pain Research Forum, but the Pain Forum, where basically the setup was that they would have somebody write a review or position paper and then invite a bunch of people to sort of write responses to it. And this was, I forget what year it was, but it would have been one of the anniversaries. It might have been in 1995. So this would have been the 30th year anniversary of the gate control theory. Is that 95, 75? Yeah, not 30th year. I can do math. And the premise that I put forward, or at least the thesis that it was myself and Michael Morgan that wrote this piece, was basically that the gate control occurs at the primary afferent. That is, that because of these systemic factors that you just raised, it isn't just that the excitability or the information moving out of the spinal cord to the brain is gated by relative activity and low threshold afferents, descending inhibition, et cetera, but that one's level of stress and the release of mediators from the sympathoadrenal axes or the hypothalamic pituitary axes, all of those are associated with the release of chemicals that act on primary afferents and influence their excitability. And so that sort of, at least in my mind, was an example of how you could have systemic effects that were influencing basically pain, but starting at the peripheral terminals. And so the short answer to your question is that I think those systemic effects have huge effects, that sort of our percept of what's going on in the world is associated with sort of, on a minute to minute, second to second level, associated with changes in sympathetic tone, relative levels of cortisol, relative levels of a whole host of mediators that influence not only how the central nervous system functions, but how the peripheral nervous system functions. And so all of those factors, I think, are key. That's why, at least in my personalized medicine approach, I think you need to, the biopsychosocial model, I think, is the relevant model of how we need to think about pain. That it's not just biology, it's not just nociceptors, it's how they're functioning in this intact system. I tend to have a very primary afferent perspective or view of the world, in that is that it being that is the primary afferent, the nociceptor in particular, is a viable therapeutic target. Not only is it dynamically modulated, not only does it contribute critically to differences in our percept of pain, but it's a good therapeutic target because you can avoid CNS side effects. People tend to focus on these centralized pain syndromes and, nosoplastic pain sort of as being, used to largely replace this verbiage for a centralized pain syndrome. I think nosoplastic should be, as intended, viewed to just say altered processing of the nervous system, and that the changes in the afferent input can be driving that, can be just as nosoplastic as any node as you move up through the system. And none of these systems function. When you put it back into a human, none function in isolation. So they're all dynamically regulated, whether it's the central terminals modulated through canonical descending inhibitory circuitry or the peripheral terminals modulated through these larger contexts. As you suggested, what is now one of the sort of most exciting and rapidly growing areas of at least pain neuroscience, if not, cancer neurobiology and everything else, is the sort of neuroimmune interactions and how the nervous system is influencing the immune system and vice versa. And I think that's just an additional layer of complexity in terms of how, the immune system playing, yet another bridge between how the CNS is functioning.
On that note, and we also talked about genetics and the influence on the personalized medicine, I want to ask you, Michael, what role does interdisciplinary collaboration combining all of these genetics, physiology, immunology, computational modeling, what are the role that all of these areas play in pushing pain research forward? Do you foresee a deepened collaboration of those areas, or do you believe that they should be a bit more disentangled, each one finding their way and then combining at the end?
Right. Well, the minimally they make science a whole lot more fun when you've got all those different people talking and interacting, right? They all come from different perspectives. They have different backgrounds. They view things differently. And just hearing how different people think about a problem is helpful. I find helpful to how I think about a problem. It just gives me a different window or angle to approach it. Fundamentally for these types of complex problems, I think those sorts of interactions are going to be the only way that we're going to solve them. I think for those of you that come from the purely immunology side, immunology as a profession tends to be focused on immunology, and they don't really think about other tissues and other forces. There's so much complexity just in terms of how the different immune cells are talking to each other and signaling and innate versus adaptive and so on and so forth. And you can account for so much pathology simply by focusing on the immune system that, why should I worry about the nervous system on top of that? That many get through an immunology program and they don't have the training and the language in neuroscience. And the same is really true about neuroscience, right? The neuroscientist, it's a very complex system. How one neuron talks to another. Why should I worry about immunology? But it is unequivocally clear that neurons and immune cells are talking to each other. And if neurons and immune cells are talking to each other, then neuroscientists and immunologists need to be talking. And so the people who have the training in both, I think it's fantastic and, all the power to them. But, neither field is trivial in and of itself. And so it certainly helps to have people who are, rather than expecting somebody to be an expert in both, have the two bringing those expertise together. But when you start to factor in additional issues like, how does the system work as an intact, multifactorial, complex, interactive system, having a modeler on board, having additional levels of expertise, having people who can handle working with large data becomes essential as well. One of our projects is trying to study mechanisms of neuromodulation. And having computer modelers involved who are simply thinking about how current spreads when you've got an electrode implanted in a DRG or a spinal cord is critical when you're sort of scaling between humans and rodents, for example, or just looking at human tissue of thinking about, what are the constraints on that system and therefore what might be underlying mechanisms, thinking about how that current spreads, if it isn't impacting the dorsal horn neurons directly, that has fundamental implications for how this thing might be working.
Yes, yes. Thank you for that answer. Yes, I think that each area has its contribution, and it's great that we can talk and we can see new perspectives to develop new therapies or discover new things.
If we're going to solve a problem as complex as pain, it's going to require an inter and multidisciplinary strategy. It'll be the basic scientists working with psychologists, working with healthcare providers, input from persons with lived experience. That belief of solving a problem was going to require that kind of interaction was the basis for the founding of the International Association for the Study of Pain. It's what the PRF has really been formed to do, is facilitate that dialogue and interaction. For those of you that aren't members that are listening to this, you really should be.
Switching gears to mentoring and future directions, and finally, as a leader in pain neuroscience, what advice would you give young scientists entering this field? Which skills or approaches do you believe will be most important or breakthroughs in understanding and treating pain? It is always nice to ask this question because everyone has a different perspective, and we are dying to hear yours.
Well, it's an interesting question. One of the classes that I teach here at the University of Pittsburgh is a grant writing class. And so we get to see every semester, the classes focused around the projects that each of the students are working on. And we get them in the second year of our program. So they sort of have a sense of what they're doing. And, as you go year to year, you can really see it's the latest trend, right? It's DREADS, and then it's optogenetics, and then it's RNA-seq, and now it's omics this. As the fads move through, people want to learn those techniques. They want to learn how to use and employ, and are trying to incorporate into their research programs the latest and the greatest, the techniques and approaches. From my perspective, I'm kind of a nuts and bolts kind of a person. Fundamentally, this concept that I struggle with, particularly in that class, is just what is a hypothesis and what is hypothesis testing research, as opposed to what is hypothesis generating. And I think fundamentally, we are moving faster and faster towards a research community that is really good at hypothesis generating research. All of this omics stuff is, you identify a dozen targets or 100 targets that are upregulated, X-fold, or downregulated Y-fold. And, some fraction of those might be causally linked. They might not just be associated. And so describing those changes is simply the beginning, right? That's the first step. And people really focus on that, what happens if I mutate this gene? Or, what happens, what's the mouse phenotype? That's the first step. Those are the hypothesis generating experiments. The hard part is the hypothesis testing experiments. What do I need to do to actually establish a causal link between this, that? So from a mentoring perspective, for me, fundamentally, I want my trainees to understand at a gut level the difference between those and be thinking about the experiments that they're doing to address that. I think our clinical colleagues, certainly here in the U.S., I can't throw everybody else under the bus, but certainly here in the U.S., they sort of given up on this idea that there is hypothesis testing research. It's, they state it as, we hypothesize that if I give this drug to this patient population, they will get better, or, this pain will go away. And, that's a prediction. That isn't a hypothesis. You got to get to the more fundamental, what do you think is actually going on type of an issue. And I think just taking that extra step changes your thinking about the problem, and it changes how you approach it. Again, not to be critical of where we are, you sort of threw out the bait of, are we learning too much about the C57 mouse and not the bigger complexity of pain? There's a group in Texas here in the U.S. that runs the Gulf Coast Consortium. And they have pain researchers from around the country that give talks, but it's run largely out of Texas. And there was people that were talking today, doing some really sophisticated work with rodents and trying to assess differences in behavior and differences in social interactions and so on and so forth. But they were using this term pain to describe what these rodents were, quote unquote, experiencing. And I'm sort of, do we know? And I would argue, in the most extreme case, that the rodents really don't have enough of a cortex to really experience pain the way a human experiences pain. So, to the basic question of do rodents feel pain, I would pose the heretical concept that, in fact, no, they don't experience pain. They experience nociception, but pain and suffering that humans experience, I think, is something that rodents don't necessarily experience. And, again, now we're getting into potentially philosophy. But I think as scientists, we need to be very careful about the terminology we use. And as scientists, the precision of our words, I think, is critical. And so, what we're talking about when an animal is now withdrawing at a lower threshold, is that pain, or is that hypervigilance, or is that hyperexcitability in the dorsal horn that doesn't really do anything to the cortex? I have a little video poking my cat's tail with the pen, and I'm barely touching the tail, and the cat's flicking its tail away. Is that pain? No. Right? Is it even hypervigilance? Well, yeah, I mean, the cat knows that I'm kind of messing with it a little bit. I wouldn't call it pain. I wouldn't even call it hyperreflexia at that point. And so it's easy for us as scientists, particularly when we work with preclinical models, to want to anthropomorphize, to make it simpler to talk about pain behavior. But I think we need to be careful about that because it influences our thinking and it influences our ability to translate what we're doing to approaches to treat pain. So again, long-winded answer to your question about mentoring, but to me, it's that, precision in terminology. It's thinking about experimental design. For me, fundamental nuts and bolts about how you approach a problem. I think we as a profession, or we as scientists, are potentially doing ourselves a disservice if we lose sight of those factors and, look, here's now 6 targets for the treatment of pain. Well, no, not necessarily, right? Those are 6 proteins that are changed when you damage the nervous system. None of the 6 may have anything to do with pain, let alone the excitability of the neuron. And so we need to go back and do those critical experiments. The broader question about mentoring, I think, don't forget there was people doing good work even back before the 60s, right? That pre-Twitter and X and whatever else, Blue Sky, that there was really solid work that was done that we're now replicating with more sophisticated techniques. But we're replicating those observations. We're not moving the field forward. Don't lose sight of the literature.
Thank you for sharing your story, your path in science, and your beliefs. It was very inspirational for us.
Wow, thanks for taking the time, and thanks for asking me.